I have to thank you for this invitation for a small presentation, but I think it's representative of what's going on with CFS in Germany. It is the acceptance is extremely limited, and even more limited is the amount of research being done. So we are a small group who went up ourselves to get forward. I'm working more clinically in an immunology clinic in Munich, and I have been engaged in some or the other way in CFS for now more than 10 years. Professor Kramer, he's from the Immunology Institute in Heidelberg. He is more collecting the data, and when I talk Professor Wallich from the Institute of Immunology at the University of Heidelberg, he's actually the one who is doing the research work. He is, I think, an expert for more than 20 years in molecular biology, and his major focus has been all the time Borrelia. So it is, and they are running the blood center of Heidelberg, and they have never been engaged in virus detection, which kept their rooms quite clean. I just want to go through it because I have just a few minutes very shortly. We focused on detecting XMRV in Germany too, especially since we were challenged by, or this was challenged by a work which has been published last year, and they didn't find any. So we have to thank Judy extremely much for helping us up and keeping us encouraged in getting forward with that. We developed a test system in the beginning following her protocol, but we found out that we really have to improve it. We did a lot of testing for several times of incubation. We did co-culture with culturing for up to 7 weeks. We did co-culture with susceptible cells and all that, and find out that in the beginning we didn't find any, but with the time of culturing we got more and more samples, and we, like you see here, we also try to make a calculation of how much of virus we can get. So I think really the conclusion is that the virus presence, at least in these cells, is extremely low. Whatever that means, we cannot tell that. Since we cannot go into clinical work with those results, we also focus for a longer time already, but accelerated that too on the presence of other viruses. So what you see here is what we did first according to the Lombardi protocol. We didn't see any patient. Then we improved it with nested PCR and the culture, as I told you, and we got more and more positive results. Right now we have about 100 patients under work. They are still under work, and I can't tell you how many of them are positive. Approximately 40%, but I still think there's a lot of work to be done to improve the techniques and also to extend it to other, to related virus species, which we didn't do so far. We also developed a vessel blot test recently, but this still needs some more work. Here I show what the kind of antigen we used for the test, and some samples where you can see that there are antibody positive persons among our group too. I cannot tell you how good the coincidence is of virus detection and antibody detection. We still have to wait up until we have the full workup. So we focus on EBV2 because I think among the best papers about treatment of CFS are the two reporting about antivirals used in long-term treatment of patients. So we added this to especially EBV, which we think is the most important one of them. And actually in Germany, CMV we find not quite often. Of course, we have HHV6, which we also try to look at, but it's not as prevalent as EBV. We follow this protocol where you can distinguish certain latency studies of EBV and have set up the the gene expression for these. And I just can show you some examples. Here's one of the latency, mid-time latency genes, LMP1, which we can find in CFS patients expressed more. And we also do some testing, not exclusively at a certain virus, but more at antiviral activation. And we focused in the beginning now on these two. And there's also a sample where you see that CFS patients, quite a few are positive for XMA. So this is just a very short introduction what we are doing. We are certain, and we did a lot of, so we, again, I have to say, our last co-worker, so we did a lot of work in omitting contamination. We have a very good setup at the University of Heidelberg, the Institute of Immunology, and so we are sure that XMRV is in Germany present too. We don't know what that means, but of course it is very challenging. And we also see, at least in the example of EBV, that we have a lot of infection co-activation, and as the literature shows, it might be reasonable, at least in a certain part of patients where we see the co-activation, that we do antiviral treatment directed against that. Thank you very much.
Thank you.